welcome to MAPS tonight. We've got a great talk for you. I'll introduce our speaker here in a few minutes, but a few announcements because, of course, what is a good gathering without announcements? We all have to deal with it. But I do want to ask you two questions as I go through the announcements. What has 4 legs, 2 ears, a trunk, and 4 tusks? Anybody know? It's an elephant with spare parts. Or a Gomphothere. Okay, take your pick.
Also an elephant with spare parts.
What was that?
I said also an elephant with spare parts.
An elephant with spare parts, right. What game should you never play with a Gomphothere? Squash. Okay. All right, so Science Olympiad was here just, was it 2 weeks ago now? So this is on our show, but we sponsored the Science Olympiad, the Regional Science Olympiad here at MJC, and it was actually a really, really good time. So that's something to keep in mind for the future. It's a fun event for the whole family that comes out, and it's kind of fun for people to see what's going on with that. But moving on. Didn't look at all of these slides. So there's the horticultural program, apparently. That's coming up. What was that?
It's the colloquium.
It's a colloquium. Oh, it's science colloquium. Okay, so it's a science colloquium talk. Denise is going to kill me now. So this is one of our other lecture series that we have at MJC, which is also free to the public. It's in the afternoons, and you can see Wednesdays at 3:15 to 4:15. And this is the next talk that's coming up. I used to be on that committee, but I'm not now, so I'm going to plead ignorance and move on. This is the MAPS committee. You'll see the members that are here, photos up there. Yes, I hate that picture. But anyway, we have a whole series of talks that we do throughout the year, and we try to tie them at times when people can come out on a Friday night so that you can get the talk and enjoy a little bit of science on one night a month. Also, we have one night a month on the first Friday of every month is the science nights, and those are at the Great Valley Museum. And not only is the museum open, but we also have the planetarium shows that are both astronomy, but we also have shows that are like kind of quasi three-dimensional projection videos that are also shown in there. And then, of course, we have the telescopes open up on the roof as long as weather is permitting. So if you want to get a good look, and you can see our lovely telescope up there. So if you want to get a look at that, you can. Great Valley Museum, of course, is always something we like to share with the community. It's very inexpensive. You can see prices are right here. Basically, it's $3 to $5 to get in. Students are always free. Faculty are free because we pay a fee. But anyway, so that's the Great Valley Museum. That's where the planetarium is. And then this is the next talk we have coming up on April 14th. That's going to be right here. And it's the Blow Your Mind Botany. So he'll be talking on that one coming up. And tonight, it's Stompin' Through the Marshes. And it's the Gomphotheres that she's going to be talking about tonight. But again, I got a couple more questions about Gomphotheres. Because I love Star Wars. What's the Gomphotheres' favorite Star Wars character? The Tusken Raiders. Why did the Gomphothere get made the curator of the Paleontology Museum? Because he was the multitusker.
That's a good one.
Yes, I have more. 2 more and then I'll be done, I promise. All right. What do you get when you cross a Gomphothere with a kangaroo? Giant holes in the fossil record. All right, the last one, and I had to kind of get a little kibitzing on this a little bit because they're not quite as hairy as a mammoth, but what do you call a Gomphothere who sings calypso? Hairy Elephante. Yes, it is. All right, I'll quit with that. Okay, so Dr. Jeanette Pirlo. She is the brand new evolutionary biology assistant professor at Stan State, and I say brand new, literally she's in her 2nd semester. Her research focuses on paleoecosystem reconstructions through mega-herbivore interactions, diet reconstruction, and population dynamics. Additionally, Dr. Pirlo is committed to broadening representation of STEM through support, mentorship, and inclusion inclusion of students from non-traditional backgrounds in historically excluded communities in research. In fact, she's got an internship program that we're talking about here at MJC, so stay tuned. Anyway, a native of the Central Valley of California. She grew up in Manteca and Riverbank. And in fact, her mom and was a sister and brother-in-law are here tonight. She graduated with an AA in biology from Monterey Peninsula College, a Bachelor of Science in Marine Biology from UC Santa Cruz, and her PhD is in biology with an emphasis in vertebrate paleontology, and that's from University of Florida. I should say go Gators.
Sure.
Okay. And her PhD was done with, in cooperation with the Florida Museum of Natural History. Her research program at Stan State looks to reinvigorate historical paleontological research of the Central Valley and sharing local communities, increasing awareness and engagement of the Central Valley and their geologic history. And Dr. Pirlo is developing a rigorous museum studies and paleontology program at Stan State, and looks forward to growing research opportunities for undergraduate and graduate students interested in studying paleontological history of our community. In her spare time, Dr. Pirlo enjoys weight training, hiking, and catching up on missed naps. Her words, not mine.
It's true.
So without further ado, I want to introduce to you Dr. Jeanette Pirlo.
All right.
As I figure out how to shut the screen off.
And I figure out these mics. What would a talk be with professors that don't know how to use technology, right? Actually, maybe I don't know. Is this one on then? Yeah. This one too? Okay. The green light's on, so does that mean it's on? All right. We're good to go.
I'm just going to turn this center. Problem is, I don't know how this particular machine works.
It's off now.
Yeah, I just didn't turn.
Maybe not.
Now it's going to turn blue.
System off?
So projector, center, and power off, and let's hope I don't recycle everything. There we go.
All right. Thank you for that introduction. I really appreciate it. And thank you, everyone, for coming tonight. I appreciate y'all being here on a Friday evening. I'm sure there are many more exciting things to do, except that we're talking about Gomphotheres. And how many people know about Gomphotheres that are not my students or my family? One person, two people, fantastic. All right, so hopefully after this, I introduce you to the wonderful world that are Gomphotheres, and we'll grow to learn, love, and appreciate them, because we often only hear about mastodons and mammoths, right? And obviously now modern elephants. So we'll go and stomp through the marshes of Florida to look at the Gomphotheres from that area where I did my PhD research. I'll do a quick introduction, species description, and some paleontological reconstructions, as well as discuss the population dynamics of the site and some of my future research that I hope to do over the next couple decades of my professorship. To say out loud is a little daunting and shocking. All right, so a little bit about me, and Dave has mentioned this about me. I was born and raised right here. I was born in Tracy, grew up in Manteca, had family in Riverbank with my grandparents, so we spent a lot of time driving back and forth between the two places. I am a first-generation proud Latina, and first in my family to go to college and earn a PhD and now a professorship. Both of my parents immigrated to the U.S. here to give my sister and I a better future, and I think we have made them proud, and hopefully I don't cry because my mom's here. So I received my Bachelor of Science, as Dave mentioned, from UC Santa Cruz with a focus on marine biology, and I really thought I was going to spend the rest of my life chasing sharks. I just didn't know how I was going to do that. Unfortunately, instead of chasing sharks, I was in a paleo class at UC Santa Cruz, and I really thought paleo was just a bunch of crap, because you're really telling me the slide are the blood vessels of a T-Rex? I don't believe you, right? And so my advisor, or my professor at the time in paleo, suggested I go meet this random person that's visiting from Florida because I was doing a paper on Megalodon. And I was like, I don't know who he is. And she's like, he's buying lunch at the fancy restaurant. And I'm like, I'll be there because I'm a broke college student and I've never been to the fancy restaurant, so let's go. Meanwhile there, I got talking with this gentleman who is a rock star in his own right. Nobody really cares unless you care about fossil horses. But I was talking with Dr. McFadden, and he was super excited about my interest in working with communities similar to mine, and asked me to come out to Florida to dig. And I was just like, I'm a college student, I can't really afford it. And he's like, what is this affording thing? I'm paying for everything. I was like, I'll be there. Send me the tickets. So I went to Florida, found my first fossil turtle, and lost my God-given mind. That was it. That was all I wanted to do, is find fossils, and I needed to figure out how I was going to do that, because what is my marine biology degree going to do to find fossils, right? So long story short, 7 years later, I'm now a professor here at San State teaching vertebrate paleontology. With now I can do whatever I want. I can study marine, I can study terrestrial, and so it's very exciting. And how did I get here? A little bit more. That's me and my dad. Obviously, he caught the bigger fish, but I was very proud of myself. I was always in love with animals, and so all I knew was that I wanted to work with animals. I didn't know in what capacity, and now they're really dead. So no concerns about having to keep them fed, right? But one of the main things I want people to take away from this is take opportunities. Whatever opportunity, no matter how random, out of the way, and not like what you're planning your life to be about, take it anyway, because if someone's paying, take that free trip. You never know what you're going to get out of it, or what you're going to learn, or what you're going to enjoy. So once in Florida, I was able to do a little bit of shark research. So if you're interested in Megalodon, there's a paper that came out last year where we reconstructed Megalodon using 3D images of teeth and a reconstructed vertebra. And so we reconstructed the size and shape, and it was led by a graduate student now by the name of Jack Cooper, who's doing amazing things in shark research, so he's fantastic. Then I fell into elephants, literally into a pit full of elephants. My idea was to do some fossil shark research for my PhD. I was going to save modern sharks by looking at the fossil record, figuring out where they were in the past, and then figuring out what their potential boundaries were going to be in the future. My advisor thought that was boring and that sharks were no longer cool. And I was like, what am I going to do now? And he's like, what about this dig site that you started your career in? What about the dig site? What do you find interesting there? And one of the most fascinating things is these giant elephant and rhino teeth that are multicolored. The ones that we find still in the teeth that were already functional were one color, and the ones that had not came in, that were still developing, were a completely different color. And me not having a background in geology, I didn't understand the difference, why that was happening. And my now retired collections manager, who's a dear, dear friend, was just like, don't you know anything about geology? And I'm like, no. I know about oceans. I can tell you why there's currents, but I know nothing about rocks. So from that, seeing these really cool teeth that look nothing like what I thought an elephant tooth would look like, that was different colors, that apparently was telling me where in the tooth, where in the jaw this tooth was located, it had ever erupted. Now this was what I was going to study. But I had no background on terrestrial animals. All I knew were about really big fishes with triangles for teeth. So I had to cram a lot and eventually got through my dissertation, luckily, and now I find myself here and I get to share that. But I did have this unconventional path, and this goes along with my second theme for the night. So not only is it taking opportunities, but also accept the unconventional path, because you don't know where it's going to lead. I started my career as an academic at CSUMB right out of high school, did not find academic success there, did not have the support that I needed to find success as a first-generation student. So instead of, and this is the first time my mom's hearing this, instead of me dropping out of college and coming home with my tail between my legs, I told her everything was great and enrolled at the community college in town. Sorry, Mom, I lied. And so instead, I went to community college, and there was a lot of shame involved with that, right? Like, oftentimes in high school, we're told that if you don't go straight into a four-year college, you're not smart enough, you're not going to do well. But the reality is, I found so much more academic success at MPC, and I also learned so much more about what it was that I wanted to do and the people that were around me and the type of mentor that I wanted to be, that I was able to, after several years of working full-time, transfer from MPC to UC Santa Cruz. And along the way, there was a lot of failure. That's why that circle is huge. I could not, for the life of me, pass pre-calculus. I kept failing and failing and failing, and I failed pre-calculus 5 times. At the 6th try, if I didn't pass it, I couldn't take it anywhere else, and that was the end of my academic career. So I had to pass this whether I wanted to or not, right? Like, it was do or die. Luckily, my instructor at the time heard my story. I talked to him about it, and he was just like, all right, let's get you figured out, and worked with me to make sure that I passed. And when I got that B in pre-calculus, it felt like getting my PhD. Little did I know I would eventually get my PhD, and it felt just the same. But I knew that if I could pass pre-calculus, I could do anything that I set my mind to, because I kept failing. So this failure taught me that I could do anything, right? I didn't know how I was going to accomplish it, but I knew that I could do it as long as I could figure it out. So I transferred to UC Santa Cruz, I met Bruce, I moved to Florida, started now studying elephants and paleo things, even though I didn't like it. While there, studied paleo sharks, and then again fell into these elephants. And that was a whole heck of a lot of fun, because who decided to pick the biggest animals at the site to study that I can't conveniently bring home to look at outside of office hours, right? Like, I could have studied rodents, carried my entire study species in a box and just taken it with me. No, I had to pick elephants, right? Like, really pick the big ones. One skull in a jacket where we cover it up with plaster to bring it from the field into the lab. We're talking about something weighing 800 pounds filled with dirt, right? Like, this isn't something I can easily put in my car and bring home. However, I would bring them home. So my partner was unimpressed with the elephant on the kitchen table, but, you know, COVID brought us to very specific situations where now I had elephants in my house, elephants in the back of my car. People are like, what do you mean you have an elephant? I'm like, don't worry about it, just go with it. Long story short, I finished my research, and now I find myself here. So that's just a little background about me, and you're not here to hear about me, you're here to hear about the elephants. So let's get into that. A little point of reference where we are in geologic time for this research. We're in the late Miocene, right at the cusp of it switching over to the Pliocene. So this is about 6 to 5 million years ago in north central Florida. And if you know the geologic history of Florida, it has been underwater multiple times, and it has been above water multiple times. Florida does not have dinosaur fossils because during the Cretaceous, during the Jurassic, in the Mesozoic, Florida was underwater. So could there possibly be dinosaur fossils somewhere in Florida? Sure. But they're probably under a lot of dirt, and no one has dug that deep to find them. However, we do have a lot of fossils from much more recent times, especially in the Cenozoic, including some of the Eocene. But importantly for me, Florida is kind of central for Miocene and Pleistocene research. In regards to Proboscidean evolution, right now the only Proboscideans that we know are which ones? That are alive. That are alive. Asian elephant, which else? And is there only one type of African elephant? The answer is no. There are 2 African elephant species. There's a forest elephant and the savanna elephant. So there are 3 elephant species alive now. However, through geologic history, there have been many elephant species types, or at least related to elephants. The ones that we know the best are mastodons and mammoths. We also have the Gomphotheres. But I do want to point out Dinotherium here on the left It was thought at first that Dinotherium's tusks were put in incorrectly by the Russians who discovered this in Russia because they were weirdly downcurved, and people made fun of them like, oh, those crazy Russians. They put in those tusks wrong. Like, clearly, Dinotherium had tusks coming up, just like we see with the other elephants, right? Those tusks must have come out of its upper jaws, not its lower jaws, crazy people. And then we found a Dinotherium skull with jaws and the tusks still attached, and we realized the Russians were not as crazy as we thought, and they'd actually placed them correctly. But they're a very unique group with lower tusks coming out of their jaws and kind of downturned. So we thought that they were used more for digging down into the sediments. Mammoths, elephants, modern elephants, and Gomphotheriums all have upper tusks, but then the funky group of Gomphotheres also have lower tusks coming out of their lower jaw, and some mastodons, early mastodons, also have lower tusks in their jaws. So how did elephants get into North America? Much like many other species that evolved in Africa, Asia, they followed the corridors of land as they opened up, and they found those resources that were applicable to them. Mind you, animals aren't wandering around saying, you know what, I really want to go to North America. They're really just walking around finding the resources that are appropriate for them and moving into those areas. Eventually, getting stuck there when conditions change and that corridor between Asia and North America seals up when Beringia goes back underwater. But elephants arrived in North America twice in two pulses. Once at 16.5 million years ago, and again with the Gomphotheres at 15 million years ago. And they came down, like when we think about the Pleistocene with the mammoths, with human migration into North America, down from into the west coast, eventually crossing the entire North American continent. You can find elephants all across North America, and once the Isthmus of Panama arose, joining South America with North America, two species went down into South America and became endemic to that area. So elephants have a global history except Australia and Antarctica, but I am dead set on going to Antarctica, so I need to figure out if an elephant swam across the ocean, right? Probably not, but I'm going to work on that grant. Okay, so the map on the slide is in regards to mammoth movements into North America, but it works great with all migration of elephants into North America, as they all evolved in Africa, are considered Afrotheres, and spread out north through Europe, Asia, and into North America, and eventually south. Mammoths did not make it into South America. That's why you don't see the arrows extending into South America, but other elephants did. And for point of reference, I work here with specifically Rhynchotherium, which is related to Gomphotheres. And it's thought that Rhynchotherium gave rise to Cuvironeus and Nodiomastodon, which are the endemic elephant species of South America. Looking at their teeth, so these are all elephant teeth of different types. Do they all look the same? Would you have known that's an elephant tooth? Any of them? No, right? They have these weird teeth that look nothing like you would imagine a tooth looks like, and you can't tell because there isn't a scale on there. But an adult last molar is about this size. So it's about a foot in length for all elephants. And elephants are really unique. The tooth shape is adapted to their feeding preferences. So the ones that have more of a flat plate, like the mammoth and the African and Asian elephants, they have more of a flat plate. Those teeth are used to grind up grasses, which are much more abrasive. So they need very tall teeth that can handle that abrasive material and not go toothless by the time they're two. Whereas the mastodon and the Gomphotheres have much more bulbous teeth. They look very different from the mammoth tooth below. And those teeth are used for crushing soft leaves and branches and fruit. So if you think about our teeth, we have a lot of cusps, and we use those to crush food in between. We don't have the teeth like horses or cows that grind their teeth to grind that plant material. So looking at the tooth shape, we can determine the diet of animals. One important thing to note with elephants is that they have horizontal tooth replacement. So unlike most mammals that have vertical tooth replacement, where we lose our milk teeth and have our permanent teeth come in at young adolescent age, elephants have vertical tooth replacement like sharks, where they're coming in a conveyor-like fashion. The back tooth will come in vertically and push the front teeth forward until eventually the tooth in the front falls out. And so elephants have 6 teeth on either side of their mouth throughout their lifetime. They still have the same amount of teeth as most mammals. They're just coming in a different fashion. And so based on that, we can determine the age of an individual. Was it 5 years old? Was it 20 years old? Was it 50 years old? Based on the tooth present, because modern elephants have the same mechanism for tooth replacement. So we've been able to track the age of an elephant with the tooth present and being used at the time of that age. So we've applied those same age ideas to extinct elephants. And we can determine the age of that elephant based on the tooth that we find present in the jaw and the amount of wear. So here in this Gomphothere picture, this white part that you see is the dentine of the tooth, with that caramel colored portion being the enamel. And if you look at this back tooth, here you can also see the difference in color. It has that dark chocolate to that caramel color. This dark chocolate color is pertaining to the teeth that have not yet erupted fully from the jaw and is still forming. But you can see, you don't see any of that dentine, that trefoil pattern of wear on that tooth. So if I was to find the unworn tooth somewhere by itself, not in a jaw, I can say this is whichever molar it turns out to be. Say it's the second molar, the M2, and there's no wear. So this came from an elephant that had already created this tooth. It must have been somewhere between 30 to 40 years based on our estimates, but that tooth fell out of the jaw because it has no wear on it. However, if I was to find this tooth that has wear, and if the first one is the M2, this one is the M1, the first permanent molar, because it has wear on it, I can say this tooth was on the jaw that was actually crushing food up, and it's probably from an individual that's around 20 to 30 years old, based on our estimates and based on the amount of wear present. In addition to this, we can also determine what they're eating, not just by the shape of their teeth, but also by the chemicals found in their teeth, even though they are fossils. So for the most part, organic material does not survive past 10,000 years or so, so we can't really extract any DNA or do carbon-14 dating on these fossils that are 5 to 6 million years ago. So how do we determine the chemical composition? Shouldn't that organic material have dissolved away just like DNA? Except that carbon in the form of a stable isotope or oxygen is not going to disintegrate in the same fashion as DNA or other proteins. So that is locked away into the matrix of the enamel of the tooth, into the actual tooth structure. So if I collect the sample of that tooth, of that enamel, I can run it through a mass spectrometer and figure out what the carbon-13 to carbon-12 to carbon-13 ratio is, and determine, based on our understanding of modern plants, what it was eating. So C3 plants, those trees, shrubs, bamboos, forbs, and some grasses normally grow in temperate and tropical areas. And they collect sunlight differently and arrange that carbon from photosynthesis in a different manner than C4 grasses, which are your typical grasses that you see, the grasses that you'd find at a golf course. Those are C4 grasses. So their chemical structure, the manner in which they arrange carbon, is different from each other. So because of that, we can take the enamel from these teeth, grind it up, run it through a mass spectrometer, and figure out what the ratio of carbon-12 to carbon-13 is, and determine if these animals were eating C3 grasses or C3 plants or C4 plants, or if there were some type of mixed feeder in between. However, for those of you that are in or have taken biology, are C3 grasses or C3 plants and C4 plants the only plant types there are? No, what am I missing? Cam, right? So where is Cam falling into this? I can tell you that it's falling right in the middle, that we call mixed feeders. So Cam is one of those things that we're just like, we don't really know what it's doing, so we're going to kind of ignore it if this animal was not found in a desert area where you normally find cam-like plants, right? So mainly you find cam in succulents. If I'm finding this elephant in a rainforest, I probably don't have cam there, right? So if I get that mixed feeding signal, I can make an assumption that this animal is eating C3 and C4 and not a cam animal. If this fossil is found in a desert where there's a lot of cam plants, then I need to pay more attention to that and determine if it's cam feeding or if it really is a mixed feeder. The oxygen itself tells us how arid an environment was, how much water, how much rain was happening, but oxygen is much more difficult to interpret, so oftentimes, unless you think that there was an instability with the amount of rain in an area, most people leave oxygen off to the side. We can just say, it looked like it rained a lot, but let's look back at the carbon, right? So as I mentioned, carbon values vary based on the vegetation consumed, while the oxygen is telling us more about the amount of rainfall in that environment. Because we are what we eat, everything that we eat gets assimilated through time into our tissues. All of our tissues are being replaced, right? All of our bones get replaced every a certain amount of time. Our skin cells get replaced every X amount of time. So everything that we're eating gets replaced into our tissues. And so with teeth don't get replaced in the fashion that our skin cells get replaced, right? With our teeth, they just get replaced once. Or if you're an elephant, it gets replaced 6 times. So we can look at that carbon isotope signature and determine what that individual was eating, either a C3 or a C4 plant, and say for certain this animal lived in this type of environment and it ate these types of things, compared to another animal that either lived in that same environment and were competing for the same resources, or were not competing for the same resources, or lived somewhere completely different. Or we can say for certain the climate change and the vegetation change, right? It went from C3 to C4. So a lot can be determined from that. So the fossil site that I was located at in Florida, again, is in that Miocene boundary. And the United States, the continental United States, and into South America, there are lots of Miocene boundary locations. However, the majority of them are not found in Florida or the Southeast. We have 1 or 2 examples from that time frame, mainly the palmetto fauna, which might also be known as Bone Valley if you are a Florida fossil enthusiast, and now the Montbrook fossil site. And before I move on, I do want to mention that the Montbrook fossil site is in this section called the HH4, and paleontologists like to name things as descriptively as possible, and who knows what the Hempalian means? Only me. Why? Because we're not actually that descriptive. We're as confusing as possible to make things really difficult for people that are learning it. So we use a term called the NALMAs, or the North American Land Mammal Ages. Before we had the technology to chemically analyze and date locations, we had to figure out how old things were using the idea of superposition. Something that is at the bottom of a rock layer compared to the top of a rock layer is probably older down here than over at the top, right? So using the idea of superposition and the types of animals present, we could say this age is in the Cretaceous. These fossils are found from the Jurassic, depending on where they are. So because we are mammals, and the only thing we care about are mammals, we name them after mammals. So the North American land mammal ages are how we would age even smaller segments of things like the Miocene, right? So Miocene covers a large expanse, but if I attach land mammal ages, I can now narrow it down to a smaller area. And if you hang out in the Miocene with me and I tell you I work in the HH4, you're going to be like, yes, I know exactly what that is. For those of you that don't hang out in the HH4, that's around 4.5 to about 5.5-ish million years of age. And so that is straddling the boundary of the Miocene and the Pliocene. So on to the fossil sites. And this artwork I want to mention is made by Dr. Rachel O'Keefe, who was a grad student at the same time as I, and is a fantastic artist and has a book out of dragon artwork that she recently made a couple of years ago. Anyway, so the Monbrook fossil site is found in northeastern Florida, north central Florida, and it's about an hour north of Orlando. So if you've been to Florida or you know where Orlando is, it's right smack in the middle of the state. I always thought it was near the water because I assumed everything was by the water in Florida. It is not. It is in the middle of the state. So Monbrook is just an hour north of Orlando. So you can go and dig. They do accept volunteers. So if you find yourself going to Florida and the dig site's open, you can volunteer and go dig some elephants and then spend the weekend at Disney World. It's very convenient. So this site, it's believed to be an ancient river that cut through a forested environment. And we think it's a river not only based on the marks here, the sediment, the stratigraphy here, but also because of the animals that we find. There's lots of fishes and turtles, including a new snapping turtle that will be described soon by a friend. But we also know it's a terrestrial site based on the animals present, including my elephants, rhinos, saber-toothed cats, and some other animals that I'll discuss. So this is a fairly productive site. There's lots of fishes. We have marine fishes as well as freshwater fishes. So we know that the site was either near water, near the marine water, or that it was getting some marine incursion during different times of its period. The site was first discovered in 2015, and I love this story. This was my favorite story to tell when I was at the dig site with our new volunteers. It was discovered in 2015 by the 4-year-old granddaughter of the landowner. That's Forrest right there in the corner. She's now no longer 4. I'll let you do the math. But she was dead set on finding dinosaur fossils on her grandpa's land. He mines the sand in his land to sell for building roads and building new school buildings. And so nobody wanted to tell her that she wasn't going to find fossil dinosaurs, because remember, Florida's underwater during the time of dinosaurs. So she jumps into one of the pits and she comes out with bones. And she's like, I found a dinosaur. And her grandma and her mom are like, oh my God, she found a dinosaur, you know? And so there was concern that perhaps it was ancient human remains. And if that was the case, then they'd have to cease operations, financial operations, and let archaeologists come in and study that and collect any specimens. Eventually, the fossils made it to us. We determined they were not human remains because that's a big old toe at the top. I don't know any humans with that size toe. But it turns out that she found the toe of an elephant and several turtle and fish bits that you see up there. And our collections manager, whom I mentioned, thought I was dumb and knew nothing about anything, and he wasn't wrong, but he also was not right, is a very stoic individual, doesn't get excited about anything. You know, you can tell him a joke, and he's like, all right. Like you're wasting his time. My understanding, this happened before I got there, but because it was such a big event in Richard's life, saw these fossils, got excited, went straight to the dig site the next day, and we have been digging at Monbrook since 2015, fall of 2015. Operation ceased a little bit during COVID, but we would sneak out every couple weeks to make sure that the site was still okay, because in Florida it rains all the time, so we need to make sure that the fossils that we had found were covered up and we're not getting damaged by any additional sand flowing in. But for the most part, we have been digging nonstop since 2015. We do about 2 seasons a year in the spring and the fall. The majority of the workload is completed by our community volunteers. We've probably had over 2,000 volunteers since 2015, all of them being from about up to starting at 14 years old to retirees that come out and spend their time with us. And so even though it's ran by museum people, by paleontologists, the bulk of the work and the bulk of the discoveries have been done by our volunteers, as well as a lot of the cleaning, the preparation, the identification, and inputting into our catalog system. So they're amazing because through them, at least 1 million specimens have been collected at the site since 2015, with many, many more every single day we're out there. So some of these important fossil finds that have allowed us to place Montbrook within the specific NALMA, debatable still, but we know it's in the late Hempalian. Whether it's HH3 or HH4, we're still arguing about that, but give us some time while we come up with our decision. But we have found the oldest record of a swan in North America, and that was found by Cindy. And funny story with this swan, I had spent 8 hours taking down this square, this 1 meter by 1 meter square, the day before. And I must have moved at least a ton of dirt, and that's not an exaggeration. You carry 2 giant kitty litter buckets full of sand, you go dump them out. And I remember I counted the loads, and I estimated about 1,000 to 2,000 pounds of dirt that day, right? I was dead set on finding something. I did not. I had to come home for a few weeks, and the very next day, Cindy was working there, and right after that clay layer that I stopped at, she found this dang swan. And I was so mad that I had spent hours, sweated my life away, damn near gave myself a heat stroke to find nothing, and then Cindy shows up and finds the oldest record of swan in North America. However, I'm not that upset because I love Cindy, and she is much better at finding things than I am, and I would have probably not recognized that swan bone since it was early on in my digging career and would have probably crunched through it, so better her than me, but it's still a fun story. We also have an example of a hyena dog, Borophagus hilly, found by Dan here in the corner, as well as a saber-toothed cat with a saber still present, also found by one of our volunteers, Bill, and we have many a Bill in our volunteer group. So which Bill it was, it's left to be determined. I'm just kidding. It was Bill Buhai. All right. So from this information, we're able to determine that Montbrook is a river system because we have some turtles. We also have freshwater turtles, and we have some marine fishes, and we also have lots of shark teeth that have no wear on them, so we know they haven't been tumbled down from an older section upstream. We know that these had to have been recently placed. We believe it's an open canopy community based on the other animals that we find at the site. And from this, we can imply that Montbrook must have received some saltwater intrusion while it was being laid down. How much intrusion, how often, was it found next to the coast for long periods of time? That is still research that's open for anyone who's interested. So if you're interested, let me know. So my research focused on determining what species were present. So we knew there were elephants, but what kind of elephants, and describe them. So that is still in the works, thanks COVID. And as well as doing some reconstruction on the paleoecology, figuring out what the site actually looked like, what type of plants were present. Were there a lot of trees? Were there just a couple trees? Were there no trees? Niche partitioning, were they interacting with each other and competing for resources at the same time? Or were these animals able to find enough resources without competing with each other? As well as population dynamics. What did those herd structures look like? How old were these elephants? Is there more male to female at the site? What is going on here? So here's one of my favorite fossils at the site, and I did make a keychain out of this because it's beautiful. But what do we see real quick? Turtle. What else do we see? Stuck in a jaw. What is a turtle stuck in a jaw doing, and what kind of jaw is that? What do I say to y'all? It is a Gomphothere job. Good job. You are describing species. What is a turtle doing inside of a jaw of a Gomphotheres? Do Gomphotheres eat turtles? How did it get stuck, Dave? I don't need the scientific reason. You're getting better at this. So obviously there's a scientific reason that is boring and will be in the scientific literature or whatever. But the better story is we found this jaw upside down at the site. And so you're digging, and all of a sudden you come across a bone, and you're like, oh my God, I found a bone. And then one of us runs over, and we're like, oh my God, you found a bone, high five. And then we start working to uncover it more to better identify what it is. And we realize the jaw was upside down. So we can't remove it from the site to look at the other side because it's very fragile. This site is a pain because sometimes the bone is not fully solid. It's sometimes clay. So you look at it and it's just washing away right in front of you. It's depressing. So we worked to expose it enough so that we could wrap it in plaster jacket, flip it over, and bring it back to the site. Because you flip it over, you don't actually know what the other side looks like, but that's the side that you start cleaning once it's back in the lab. So we had a great volunteer that started cleaning down and eventually hit the zygomatic arches here. Not the zygomatic arches, the upper jaws here that I cannot think of at the moment because it is Friday evening. What was that? Thank you, the carotid processes, where it attaches to the skull itself. And then he kept going and started coming across some teeth, and all of a sudden he found a piece of turtle, and he's like, those darn turtles. We always find turtles at the site. Anywhere you put your screwdriver down, you're most likely hitting a trachemes. And the problem with trachemes is that we only knew about trachemes pre-Modbrook from 1 or 2 pieces. We didn't even have a full shell. And then we found the first couple of trachemes, and we're like, holy crap, this is trachemes. Like, this is what it is. This is related to those red-eared sliders. Oh my God, we have found it. We found the Holy Grail. They must be super rare, but we found them here. 6 years later, all we find is trachemes, right? Like everywhere you look down, there's a piece of trachemes. And so now we're like, not another turtle. Like, why is there so much turtle here? And then he kept cleaning and cleaning and cleaning and realized that the turtle is a complete shell literally attached to the teeth of this Gomphothere. So as Dave said, it probably washed down and got stuck, whatever. That's a boring story. My idea, after we look at the population dynamics, is that there are a ton of teenaged individuals at the site of elephants. And what we know from modern elephant life history is that the male teenagers get kicked out of the herd at 18. The herds are mainly matriarchal. They just raise the babies and keep the females to help raise the babies. But the boys get kicked out at 18. Fend for yourself, right? So what is a bunch of teenage boy elephants doing at a site? Are they good elephants? Or how are they behaving? Probably like a bunch of 18-year-olds. And for those of you that are 18 in the crowd, I'm sorry, but it's true. So, there's so many turtles at the site, and some of the turtle shells that we find have giant round, what appear to be footsteps preserved in the turtle shell. So what could these teenage elephants be doing at the site with all these turtles? Stomping on them. These elephants are menaces, right? So here we have this turtle who sacrifices himself for his brethren. How did he sacrifice himself? He jumped into the jaw of the elephant. He did some parkour. Some might even call him a ninja turtle. Yes, I have used this many times, and those are the only laughs I get most of the time. Anyway, and so this turtle jumps in to save his friend from all these terrible elephants, chokes out the elephant, sacrifices himself. We'll talk about this in kin selection later, Charles. Kills this elephant to prove a point to the other teenage boy elephants at the site, mess with these turtles, and you get choked out, right? So here we have a martyr at Montbrook. What is the moral of this story? Don't be an asshole elephant at the site as an 18-year-old. So the moral of the story, don't be a jerk. You won't get choked out by a turtle. Obviously, I can't confirm that is what actually happened. So we'll go with Dave's idea that maybe the turtle got washed down the river, got stuck in an elephant jaw, got preserved. 5 million years later, we found it, and now here we are speculating to what actually happened. But I do want to point out some really interesting features on these turtle shells here. I don't know how well you can see it, but there are some round holes here. And what do we know about freshwater sites in Florida? What type of animals do we find? Alligators. So we do have the presence of alligators at the site. We are describing 3 new species, including one from Montbrook, hopefully coming out later on in the next couple years, or year or two. But this poor this poor turtle got chomped on by an alligator at some point in its life, but it did not die. It survived because this portion of its of its shell is remodeled and was able to survive this attack and go on to save his brethren by choking out an elephant. Yeah, he got tired of being bullied, so he took out this elephant. Anyway, this is a young elephant jaw around 18 to 20 years old, so it fits perfectly with my story. But this is one of my favorite specimens. We fought about removing the turtle. I told them they were dumb, don't remove the turtle. It is the coolest thing, put it in a museum, because it brings up so many questions, right? You look at it and you're like, what is going on? And you can go crazy and come up with this idea that these turtles were saving each other, or you can go the boring route like most scientists and tell the real story. Anyway, there will be a child's book about this, more like Aesop fables, but give me some time. All right, on to the better stuff. So these proboscideans are unique for many reasons. They have 4 tusks for the most part. That's one of the main unique reasons about it. Unlike modern elephants, Gomphotheres and those really old mastodons have lower tusks coming out of their lower jaw. And tusks are modified incisors. There are second incisor. So when you think about your front teeth, it's the tooth next to it on either side of your mouth. That gets modified into a tusk and eventually grows out of the upper jaw. And for Gomphotheres and mastodons, the second incisor also grows out into lower jaws. And how did we figure this out? We looked at developing modern elephants and watched those babies developing their teeth and eventually growing out, and that's how we know. Those babies obviously survived. Okay. So most tusks are made out of dentine. So when you think about your tooth structure, the white portion of our teeth is the enamel. And then the portion underneath that will often get cavities is the dentine. It's much softer than enamel. Enamel is one of the hardest materials in the known world, which is why it's perfect for isotope analysis. But one of the really cool things about Gomphotheres is they have this beautiful band of enamel still on their tusks. Modern elephants, mammoths, and mastodons do not have that enamel band. So when we find tusks with an enamel band on it, we can say for certain this belongs to a Gomphotheres. Which Gomphotheres? Debatable. But we know that it is a Gomphotheres. So from this, this was one of the bases for my descriptions for the type of elephant present. And also that enamel band on these tusks can also be used for isotope analysis and other analyses using enamel. And some tusks can give you up to 5 years of an animal's life. These tusks are ever growing. They're being constantly, every day, they're putting on another layer so you can count the years on them like you would on tree rings. And some of the research done on mammoths have shown that one tusk can give you about 5 years life of that individual animal. So we can look at differences in seasonality during the life of this animal. Did it go through moments of drought? Did it go through periods of high nutrition? If you know for certain that this tusk belongs to a female, you can even see when that female was pregnant, because like in tree rings that have very thin bands during times of low nutrition, these enamel bands on the female tusk will also be very small because a lot of the nutrients being shuttled to the baby. So we can determine how often this female was present, how often that gestation period lasted, and if that still matches modern elephants. All right. So we have several associated jaws and skulls at the site, and by associated, I mean that they're found right next to each other. So oftentimes in the fossil record, you can't say that something is part of the same individual unless they're literally attached to each other and they're not a turtle and an elephant together, but they're the same animal, or they're found right next to each other. You can make the assumption that they're probably from the same individual. So we have several associated skulls and lower jaws from every age range, from brand new babies all the way to grandpa elephants on their very last tooth. So this first one here is a juvenile specimen. We have both the lower jaw and the upper and the skull. And from looking at it, is that what the jaw normally looks like in an animal? Or does something look funky on it? It's crooked. What else? Does it look squished? It looks like someone squished it, right? So what often happens in the fossil record, as more and more dirt gets piled onto these specimens, they get crushed by the weight of that sand. So we have to get really creative when we're looking at this and identifying features in those bones to see if that is actually a feature that is present in the living animal, or if that is a feature that appeared when that bone was being crushed, and can we use it in species description. So what we end up doing is we look at the specimen and we say, okay, it has these certain features. We compare it to known elephants that we have in the fossil record. We determine it looks the most like a Rhynchotherium. And then we go to the AMNH, compare it, the American Museum of Natural History in New York. We compare it with the known type specimen, the species of elephant that was defined as, or that individual organism that was defined as that species of elephant, and will forever be known to be that species. And we determine, yes, it is exactly that species, or like, no, maybe there's some weirdness about it, and maybe there's enough weirdness about it that we can call it something else. Interestingly enough, the type specimen for Rhynchotherium was found in the LA area, and those fossils were found in the late 1800s and shipped to New York by the New York paleontologists that found them. Unfortunately, it is defined, that species is defined by a broken up tusk. There is no skull of a type specimen. So Montbrook may be the site for a known skull for that specimen, which means we need to define it as a paratype, which means we say it's part of the type, but it's not the defining character. You know, it's kind of like the next one, but this is the one you're going to use because it has the most features. However, COVID got in the way and kept us from going to the AMNH to compare them. So this is future research. But the preliminary description is for Rhynchotherium edens, which is the Rhynchotherium found in the LA area. And so this individual is a juvenile, and we know that based on the teeth present. And although the little bit that's off to the left is not a part of the same individual, I did want to show you a representation of a baby elephant lower tusk. One, because they're darling, and two, they're about this big. And two, because it has that enamel cap here at the front. And so even though it's part of a Gomphothere, the lower tusks don't have an enamel, but modern elephants still will be born with that enamel cap at the tip of their tusks, and through time will eventually rub that down and be made up mostly of dentine. And on days when I'd get depressed about my research, because it's a really long time, I'd go into the collections and look at the baby tusk and be like, oh, they're so cute. And then remember that they died before they could become adults, and then I'd get depressed again. But they're still very cute. All right, so even though this is quite small, no more than 100 millimeters in length, compared to an adult tusk that can reach 2 meters in length, you can get a general idea of how big these animals can get. So 2 meters, think about the size of this table. All right. So this is the associated skull to that individual, and does that look anything like a skull? No, it is crushed. Like, where does the brain go? Why do you think the skull is crushed? Apart from the fact that a bunch of sediment landed on it. Why would the skull crush so much compared to the jaw?
Well, the jaw is all dense, and the skull has the brain, so it's easier. It has a lot more area to crush down.
Absolutely. So our skulls are so big that if they were dense bone, we wouldn't be able to lift our head. Now imagine an elephant. Their head is what? 5, 10, 15 times the size of ours, not including the tusks. We're just talking about the head. We're not thinking about the weight of the trunk. We're just thinking about the skull, right? Those skulls are massive. So for that animal to be able to lift its own head, it either is very, very strong, which it is, but it also needs to lighten the load of its skull. So elephant skulls are very thin, the material's very thin, and they have a lot of sinus tissue in their face as well. One, it helps them breathe better, but also it lightens the load of the skull, making it much easier to move around. However, it makes for terrible preservation, and it gets smashed to smithereens. And really the only thing that you can see is the palate, the top of the mouth, where the roof of the mouth, where the tongue would go, and the teeth, and also the zygomatic arch, those cheekbones. And we should have enough diagnostic features for us to determine the exact species. Here we have an adult lower jaw. We haven't found an adult skull. We have found those teenaged individuals, but we haven't found a full adult skull. But we do have skulls from super babies to teenagers to maybe kind of like middle age, not full-grown age. So we have a good ontogenetic record, or from baby to young, a good age record for these individuals. And what I really like about this jaw is, one, the lower tusk is still in place. One of those lower tusks is still in place. But also the very last molar, that M3, is in place, and it's massive. It's about 1 foot in length, and it's very worn down. So this elephant lived a very long life, 60 to 80 years, and was able to find enough food to support it. And there's still some areas in the very back of that molar that are unworn, so it could have still survived much longer. However, something could have happened that could have caused this elephant to die and get preserved. Either it was old and sick, or it got washed down during a storm. Something must have happened for it to die and preserve, and we were able to find it. But we know that this animal could have continued living. It had the ability to continue crushing food. One thing I want to point out, it has a very downturned symphysis here, and the symphysis is the chin, and that is a diagnostic feature of these Gomphotheres, that chin is downturned and elongated. It has a longirostrum, or long rostrum morphology, where the jaw is very long, and it also has a downturned chin. And comparing that to a mastodon, which you'll see in a little bit, that downturned chin, that angle, is diagnostic for the species, and mastodons don't have that. So we can determine that also from the shape. Here's one of those beautiful adult tusks. This is about 2 meters in length, and you can see that enamel band spirals along the length of the tusk. And the spiraling feature is also species determinant. So if we can determine the angle at which it's spiraling, and the rate at which it's spiraling is a very tight spiral, is a very long, slow spiral, this can also be diagnostic to those elephants. Next, we have family Mammotheridae, or the mastodon. So sounds more like mammoth, but it's definitely a mastodon. And if you notice here with that lower jaw, it doesn't have that downturned symphysis, right? The lower jaw is still long so as to accommodate the lower tusks, but you don't have that downturn in the chin. Also, you'll notice that the teeth are not bulbous, they're much more pointier, and mastodon means nipple teeth in Latin, so that's what mastodon means. And they have nipple teeth shaped teeth, so that is your fun fact for the evening. All right, so this is the lower jaw of a baby mastodon. We do have both babies and adult mastodons at the site. So now we have 2 large proboscideans at the site, the Gomphothere and the mastodon, and there's babies and adults. What do we notice about this jaw? Is it as compressed and awkward as the first one? Why do you think this one's more 3D and more looking like it would in a jaw? Do you think it's because of the type of animal, or do you think it's the preservation of the site, the geology? The geology, pretty much. So depending on how hardened up this jaw was before it got crushed under heavy sand is how well it gets preserved. Also, how did it land in the riverbank? Was it on its side? Was it down? That would also keep it stabilized and keep it from getting crushed. So even though we haven't found a lower tusk in place in the jaws of a mastodon, they do have the alveoli, or the opening where the tusk would come out. So we know that these individuals had them. And here's that skull of the baby mastodon, quite crushed. We may not be able to get enough features off of it, but we definitely know it's a mastodon based on its teeth. And finally, this is our oldest adult mastodon at the site. It's not full grown yet. It's still teenage size. So we know that mastodons and gomphotheres, at least there were teenage individuals at the site, with gomphotheres having older individuals there at the site. And this one also does not have the lower tusk in place, or excuse me, this one does have the lower tusk in place here, but it's fragmented. We don't have the full length tusk of a mastodon from the site yet. This one has its final molar, so this is more of a middle-aged individual, probably around 30 to 40, middle-aged for elephants, not for humans. I don't want to call myself middle-aged, but that last tooth, the M3, is still in the crypt. It's still in the back of the lower jaw. So even though we can see it, we can't access it yet for features, for characteristics, or for isotope analysis, because we would have to cut the jaw open to access the tooth. For imaging, if we have access to a large enough CT scanner, like a cattle CT scanner, one found at a large vet facility, we could technically CT scan this and look at the internal morphology of that tooth without damaging the jaw. So what did we learn from these elephants here? We know that there's at least two families. We know that there's gomphotheres and mastodons present. We can describe those taxa now. We have enough individuals that we can describe how different they are from each other. Again, those challenges with the description, COVID got in the way and we're unable to access the type specimens. So I can't say with a definitive answer that this is exactly Rhynchotherium edens, but the Florida Museum has a good record of Rhynchotherium individuals, so I compared it to the ones we had in-house. Same thing with the mastodon. We believe it's Mammoth matthewi, but again, we don't have access to the type specimens. And now Richard has retired, and I just started a new job, so we are discussing when we're going on our New York adventure, so stay tuned. So what are some implications from this? What do you think it means that we have these two massive animals at the site? Lots of food. Why is that? Support them, right? Elephants need a whole heck of a lot of food. And I showed you at least 4 individuals, and there's at least 40 more. So this site has a lot of individuals. There must be a lot of food present for them to be able to sustain. And I'm just talking about the elephants now. I didn't tell you about the rhinos, all the llamas, all of the horses, and all of the other herbivores, deer and tapirs that we find at the site. So there must be a very high productive area for these animals to live. So let's look at the niche partitioning. How are these elephants thus sustaining each other without coming into conflict and causing one to go extinct over the other? And that is through isotope analysis, and that was really the fun part of this study, not just the digging part, but I got to drill into teeth, and so I lived my dentist's dream. So yes, I am a paleodontist. No, I cannot fill your cavities, but I can make them bigger. So what we would do is we would take an enamel sample from the length of the tooth and do a bulk sample. So if I take the full length of the tooth and take about 5 milligrams of that enamel sample, I can find an average value for the carbon of the individual in its entire lifetime, or for the formation of that tooth. I can also do a serial sample where I can take small bits of enamel along the length of this tooth, and like I mentioned with tree rings, teeth are also formed incrementally. So the oldest formed portion of this tooth would be here at the crown, with the most recent formed portion of the tooth at the root. So if I compare the values of one individual, I can see if anything's changed, if their diet changed throughout their lifetime of that tooth formation. If I have access to the entire row of teeth, I can see if diet changed from the oldest tooth in the mouth to the youngest tooth in the mouth. So we can get a lot of information from these teeth. And I took very tiny little samples, just 5 milligrams here, that was enough. Sometimes I'm in there scraping, because a tooth is in a very awkward position and you're hoping that you don't accidentally sneeze and blow your tiny little bit of sample away, especially with the deer that have super tiny teeth and really thin enamel. Elephants have really thick enamel, so you're just in there, like gangbusters, just like taking enamel. But some of them were really delicate and hard to get to, so lots of crying, lots of TV shows to stay focused. Lots of crying. I even splashed hydrogen peroxide in my eye. It It was dramatic. I can tell you that story later. All right. So as I mentioned, the palmetto fauna is closest in age to Montbrook, and that is our most well-known site in Florida. And the palmetto fauna ranges about 20 square kilometers of Florida, so it's not just one site. So we call it the fauna because we can't call it a specific area as it covers so much of Florida. But the palmetto, or as my acronyms say up there, PFBV, if you have questions, that's the palmetto, is about 4.5 to 4 million years old, so a little bit younger than Montbrook. We have 3 elephant taxa from Bone Valley, including the mastodon, but 2 types of Gomphotheres, Gomphotheres simplicitans and Rhynchotherium edens. Simplicitans does not have an enamel, a spiraling band of enamel on its tusks, whereas edens does have that enamel spiral. From the Bone Valley, I sampled 170 bulk samples, so that's 170 individual teeth. And from Montbrook, I sampled a lot more. So in total, I sampled about 400 teeth over the span of a couple weeks and spent a lot of time processing them. You have to wash them in hydrogen peroxide and watch them spin down in a centrifuge, and they have to sit in the hydrogen peroxide for at least an hour. Our centrifuge only holds 24 samples at a time, so I will let you do the math. I also kept track of how long this took by all the TV shows and movies I watched. So if you want to talk about the Clone Wars at any time, let me know. I watched everything. Everything. Got my money's worth out of Netflix. All right. So from this information, we realized that mastodons are much heavier browsers. They spend more time eating the browse material, and Gomphotheres are spending more time eating a mixed feeding diet. So Gomphotheres have a much more plastic diet. They're able to eat some browse, they're able to eat grass, so they're not in direct competition with mastodons. They're able to eat whatever they find at the site. And we already knew this for Gomphotheres, as there's been a lot of research in regards to this, but at least we knew that the Montbrook Gomphotheres adhered to that same rule, and we could see that they were not in direct competition with the mastodons. This trend also continues through the palmetto fauna. We see the same trend. The mastodons are much more browse. The Gomphotheres are mixed feeders. And there is some individual variation within the Montbrook proboscideans, where we see some Gomphotheres preferring grazing, where they're mainly eating mostly grass, where some are mixed feeders and some spend a little bit more time eating browse. But for the most part, we know that the mastodons are eating browse. So each individual elephant has a preference in diet, much like us. They also have a preference on which side of the jaw they chew on. You can see that based on the amount of wear, so that was fun. But based on these results, I wanted to know if there were other implications with the other herbivores at the site. Were these elephants then competing with other herbivores since they're eating up all of the type of vegetation at the site, right? So I wanted to know the niche partitioning among all the herbivores, so I sampled some more and spent some more nights in the lab, and people would leave and say goodnight, and then they'd come back at 7 in the morning and say good morning. I'm still there. Still watching some TV show. Ask me how long it took. I will give you numbers. So we know with the Montbrook sites that it's HH4, but essentially sampled all of the herbivores at the Montbrook site, as well as matching herbivores at different sites in Florida of the same age and of younger ages to see if there had been any change once the elephants arrived in Florida, if those herbivores had to change their diet. I do ask no pictures, as some of this stuff hasn't been published yet, so just specifically with this stuff. So we see normally tapirs. If you know what a tapir is, they normally live in closed canopied forest environments, and thus are usually your lowest values for browsers. We do have an example of a tapir at the site. It's a limb bone. So I didn't have a tooth to sample, and so I didn't know what I was going to do. Here I am. I'm supposed to create a baseline for my browsers, and I don't have my browser. It's still there, but I don't have a tooth, so I was disappointed. But I decided that maybe the deer were enough for me to determine a baseline. Deer are mostly browsers, they're eating leaves, they're not really eating grasses, so I went to the next best thing, which I thought were my deer. However, if you look at these values, here we have the deer are here, pretty negative, they're pretty low. The further you are to the left, the more browse, the further you are to the right, the more graze. So here the deers are pretty browse. And then when we look at the mastodons, all of a sudden, what are the mastodons more browsing than the deer? So I assumed that the deer would be my baseline for my browse. Turns out the mastodons were the baseline for my browse, and so I saved myself with that. But if we look again at the Gomphotheres here, they're all over the place, right? They cover that entire spectrum of the type of food that they're eating. And if we look down here on the bottom left, the rhinos also cover that same spectrum of time. So I'm going to speed up here because I talk a lot. All right. So I do want to say that the oxygen remains stable through time, and that's for future research. But mainly we know that the herbivores were not really competing with each other, but there is that weird interaction between the Gomphotheres and the rhinos. They're eating the same thing. So I wanted to know, was there any competition between them? So I did some population dynamics. Here's some more jaws. So one of the questions was, we needed to figure out how Montauk was actually created. Was it attritional? Was it a slow buildup of accumulation over time? Or was it catastrophic? Did something major happen, a meteor land and kill everything off at once? Did a volcano erupt like it did at Ashfall in Nebraska and kill off all the rhinos, which is this image here. Look up Ashfall rhino site in Nebraska. It's fascinating. But I needed to figure out what was going on with Montauk. How was that accumulated? And based on our herd structure at Ashfall, because there was a lot of babies and right next to the moms, and there was only one male present, we assumed that it is a catastrophic event. This volcanic eruption killed off all these rhinos at once and buried them all together. So we, one, know the herd structure, and two, we see how tragically they died. Some of them are kind of like contorted and look in pain. But Montauk, we don't have that situation. We have a slow accumulation. We have an attritional accumulation based on the animals that we have on site. And as I mentioned, we can determine the age of an individual based on the tooth present and the amount of wear. So I did that with the elephants to determine the age structure. And again, we had a lot of those juvenile young adults around 18 to 20 years old. So why is that the case, right? And I already kind of gave you a kicker for that one. From there, I wanted to determine the size, because if they are competing with the rhinos, are rhinos and elephants the same size? But were they the same size in the fossil record? Maybe, right? And so we know that Gomphotheres are shorter than modern elephants, and we're a little bit longer. They're kind of shaped like hippos. The rhinos at the time, Teleoceras, are also kind of shaped like hippos. So the question was, are they competing with each other since they're eating the same thing? And interestingly enough, Teleoceras goes extinct at the Bone Valley. So 4 million years later, that rhino goes extinct. So were these Gomphotheres competing with them and pushing them towards extinction, out competing with them for that food? So to determine that, I needed to figure out how big they were. Usually you use the size of the skull or the teeth to determine the size of an animal. Unfortunately, with elephants, their teeth get bigger as the new one comes in. So it keeps telling me, like, one tooth will tell me it's 10 pounds. The other tooth is going to tell me it's about 50,000 tons, right? So what size is this animal? I don't know. Depends on which tooth I use. So instead of using teeth, I went through, used the ankle bone called the astragalus, which has been used in monkeys to determine mass. And because the ankle bone holds the majority of our weight, it should, for the most part, give us a general idea of mass if I take the area. So I did that. I took the area of all the astragali in our collection, which sounds impressive, but there was like 10. So not as impressive as the teeth. And from that, I calculated the area, and I determined that our mid-sized elephants, those teenage-sized elephants, were the same mass as the rhinos. So now my next question is, Did all of these teen-sized elephants play a role in rhinos not finding enough food? So that is future research that we're developing with my colleagues in Florida. So we found out that Montauk was attritionally created. We came up with a new way to estimate body mass, so that was really exciting. And we also figured out the age structure of the individuals at the sites. So overall, for the research at the site, we realized we had a great series, ontogenetic series of all ages of elephants. We were able to interpret their stable isotopes and determine what they were eating in the southeast. And we realized that modern niche space is similar to 5 million years ago. Those same interactions that we see with elephants and rhinos in Africa and all other herbivores is similar and still holds up to what we saw 5 million years ago. There may be some interspecific competition between two species of animals competing for the same food. That's left to be determined. And we have a brand new site with brand new specimens. So that's the novel collections left to answer these questions. Another great art piece by Rob Soto, who drew the elephant from another talk that I did. So some future research here. We'll be digitizing historic collections out at UC Berkeley for research integration. A lot of these specimens were collected in the late 1800s, but are not available for research unless you go to Berkeley to do the research. And if you live around the world, you probably don't have time or money or access to get to Berkeley with enough time to study those. So the idea is to digitize them, make 3D models of them, and make them available for further research. I, again, want to study that interspecific competition between the megaherbivores at Montbrook, between the elephants and the rhinos, and look at continent-wide movements of these megaherbivores. Did these elephants only stay within Florida, or were they moving across different areas? What about the Rhynchotherium here in the LA basin? Are they only regional to LA, or can we find Rhynchotherium up and down California? I'd like to forecast some range shift due to climate change and marine predators. My original idea, I will eventually get to it. And assess the perception of evolution and climate change through community science engagement in paleo, and increase identity formation through access to collections and by assessing the impact of our mentors on our students. So not only am I interested in paleo, as Dave mentioned, I'm also interested in increasing the science identity of our students here and helping more students find their dreams and pursue those dreams in STEM, whether it's paleo, whether it's something else that isn't as cool as paleo. I'm still here cheering you on. All right. And with that, there are many, many people I want to thank, but mainly our volunteers and everybody else up here. So if there's any questions, I'm happy to answer them. And thank you for listening to all of my rambling stories.
Thank you very much for coming.
Yeah, of course.
That was absolutely fantastic.
Thank you.
The way we usually handle this is we give those that have to go a chance to go ahead and go right now, and then we'll do questions after that. But I was, during your talk, I was thinking, what did the 18-year-old mammoth say to another 18-year-old mammoth?
Oh, God.
Or I should say Gomphothere say to another 18-year-old Gomphothere.
How was your turtle?
My mouth is full of turtle.
I knew it had to do with
I
turtle.
have to work on the delivery of that, but I think it's good. So those of you that have to write a report on Gomphotheres, how do you do that?
Say that again.
For those students that have to write a report on Gomphotheres, how do they do it?
Just look up the same resources I did and let me know because I might need some help.
Well, first they need a really big ladder.
Yeah. I thought you were serious. I take my answer back.
All right. So
Last stroke, okay.
What album
Oh,
did
God.
Gomphothere listen to all day long?
I'm scared.
Tusk by Fleetwood Mac.
That deserves an
Those of you
applause.
that was a good one.
Okay. Not as good as the
That
first
was
one, but.
pretty good. Now I think folks have cleared out. So with that, what was the lifespan?
So around 60 to 80 years old. Most elephants have the same lifespan as humans. They're pretty long-lived. Yeah. It's a great question. So with the tusks in the way, how did they get food in their mouth and down their throat? And so I didn't really talk about this, but Gomphotheres did have trunks. When we look at their skulls, they have the same location and shape of their nostrils, their nares, as modern elephants. So we can infer that the trunk is present, although the trunk is soft tissue, so it didn't preserve, and we don't have it in Florida. But we can assume that the trunk is there based on the morphology of the skull, and so the trunk helped bring food to their mouth. I have a question. That's a really good question. Yeah.
When you split the tooth open, were you able to see any chambers, like in the human tooth, there's like, pulp chambers?
So oftentimes when the tooth gets spit out, so we call them spit teeth when they get replaced and pushed out, by then the root has been reabsorbed so that they don't lose nutrients. The only thing they've lost is the calcium and everything else in the tooth, but they reabsorb the root, so you no longer see that. Oftentimes the root is no longer present, and so you only get the crown of the tooth that gets preserved. Sometimes when the tooth falls out of a jaw that's being preserved, you will see the remnants of the root that has become preserved and calcified, so that will be present. And the root is only found very low in the crown. It's not found as high up as we see in humans. So you only see the root down at the bottom of the tooth itself. So you don't have as much of a large cavity.
Is that the same for the tusks?
The tusks do have a pulp cavity towards the back, but you don't see the pulp cavity all the way through the length. That gets filled in with new material, and the only place that will have the blood vessels to grow that tusk will be at the pulp cavity closest to the where it anchors into the skull. I would assume if they fracture a tooth when it's in the mouth, it will become abscessed. We do see that with modern elephants, where if they do fracture a tooth, they need to have that tooth pulled if they're in captivity in zoos and stuff. But maybe. We don't have examples in the record so far from Montbrook of seeing like remodeled bone due to infection. So we don't have examples of that happening. The possibility of it happening, sure, but we don't have examples of that yet.
Yeah, when they cut the tusks off African elephants keep being poached, the pulp cavity is a problem because it goes way out.
Right. And so they usually bleed out.
Yeah, so it's actually a real problem, and you can't really cauterize it, so it gets really difficult.
Yeah. Other questions? Sure.
I have a question about for the Gomphothere. So when you said that the lower tusks are still prominent even when they're calves.
Mm-hmm.
So how exactly would the calves nurse against the mother? Would they be stabbing mom while they're nursing, or?
It's a great question. So we do expect these tusks are fairly short in the lower jaws of the babies, and the upper ones are also fairly short. And so they go through a milk tusk phase as well, where the assumption is that the milk tusk will fall out at the time of weaning, when they stop drinking mother's milk, which is usually around 2 to 5 years old, depending on the individual, same as children. Not adult children, hopefully, same as children. But the thought is that they're very short and kind of soft and not as pokey as their adult tusks. But yeah, that is a great question, and I haven't watched modern elephant babies nurse, but some of them will have like tiny little tusks. But yeah, I would say the mom's probably uncomfortable often. Yes, ma'am.
So I know that these did not live at the same time as humans, but are there any connections to humans?
There are connections to humans. So Gomphotheres, although mine did not, Gomphotheres did persist into and past the Ice Age and went extinct at the last Ice Age. So we do have examples in Mexico of Gomphotheres that existed past 8,000 years ago. So you can find them cohabiting with humans. They were used for food. We have examples of that 8,000-year-old individual that has a spear tip embedded in a bone, so obviously it was hunted. So they did serve as a food source for our early Clovis people in North America.
Wasn't there one found like in Argentina or something as well?
Yeah, so I can definitely go on this topic, but elephants, again, made it all the way to South America. following the same routes that we expect humans to have followed. Obviously, elephants came first, but they persisted fairly long past the arrival of humans in South America, and so you find them as food sources in their middens, as well as horses, but I won't get into that fun topic. But horses absolutely persisted in North America long past the time we claim they went extinct, and Europeans did not bring them all back. They were living in North Canada, so Native Americans knew about these giant animals. They were not terrified of the Europeans when they showed up on horseback. Tired of that misconception. That's a whole nother topic we can discuss another day.
Any questions?
Yeah.
I'm sorry for a
Yeah, go for it.
So all elephant species that migrated here to the New World, they're all extinct.
They're all extinct.
So what maybe could be the reason why all the original species are still alive in present day in the old world?
Yeah, that's a fantastic question. And so the thought is that because of the elephants that made it into North America were following their preferred food routes, especially with mammoths that ended up being major grazers, they were following those big savannas that opened up during the Ice Age and were able to find enough food, even though they were competing with musk oxen and bison for those grasses. There was enough grass present for these animals to persist. When the climate changed and it started warming up, those savannas got constricted further north, and it didn't help that humans were hunting them, right? But these ecosystems got constricted further north, and these animals were unable to find enough food to sustain them. There potentially were other reasons for it that we haven't discovered yet, but we think it's a change in their food source due to climate. Whereas in Africa and Asia, those food sources have remained stable. They didn't seem to be as affected during the change from the Pleistocene to modern times. However, that can change now as things are warming up. Hopefully these animals can adapt at the same rate of change if we stay out of the picture. Great question.
How long was that climate change that caused their demise?
For the Pleistocene, so the Pleistocene lasted, 10,000 to 2 million years with several glacial and interglacial periods. So I want to say that they persisted. There's some areas where they persisted, in Wrangel Island to 4,000, 6,000 years ago. So depending on where you're located is how long these proboscideans lasted. But I would say in the last million years or so, and really with the arrival of humans in North America, which keeps getting pushed back, and I don't know what the most recent pushback date is. I'm going to stick with 10,000, but for the anthropologists in the room, please correct me. As people started arriving in North America, they were definitely following their preferred prey, right? Again, people were not like, you know what, I really want to go to Florida. It sounds sunny right now. Like, people didn't know that existed, right? They're just following their prey. They're following their preferred habitats, places where they can find enough food to feed their groups of families. So I would say maybe in the last, what do you think, 25,000 years?
Depends on where you are.
Yeah. Let's say here. 50,000 years.
But then compare that with how fast we are changing the climate.
Right. So that change is very, very rapid now compared to what it was, right? If it took, say, 10,000 years to go from the last ice age to now, that's 10,000 years. But from the time of the Industrial Revolution in the 1800s to now, that's 200 years. And we have seen a tripling, doubling in temperatures compared from the Ice Age to now. So things are heating up faster. Climate change is a natural thing and does happen naturally, but not at the rate that we're seeing it happen, coupled with everything else, coupled with people accessing additional resources that natural environments need to survive. So it's not just climate change. It's also all of the other activities that humans are doing, accessing these animals for food, taking water, taking up their ecosystems. Again, I can go on and on. If you're really interested, take my biogeography class. Yes. Other questions?
Other questions?
Sure.
So I may have missed it, but so sorry if I did.
Huh?
But what was the cause for there being a bunch of juveniles buried at Monobrook?
Yeah, so I didn't really touch upon that, but the juveniles are really juveniles and the oldest are the most easily killed, right? So when you think about what a modern elephant, who is its predator, No one really, right? Other than humans, if we're just thinking about elephants in the savannah, maybe a lion every now and then, but what is the lion targeting? Is it targeting the big prime bull in the herd, in the area? No, it's going after the weakest ones, right? The newborns, the 1-year-old babies, or the really old elephants that don't have teeth or tusks to defend themselves, so they're already weakened. So the thought is that usually you would find a high aggregation of babies and oldest individuals at the site, but that's not what we see. We see this like middle 18 to 25-year-old individuals at the site. And so looking at modern elephant herd structures and knowing that these young males get kicked out of the herd, for 18 years, they have been helped by their parent, by their mother to find food. They have not really been paying attention, because I sure as heck was not paying attention where my mom was driving to take me to grandma's house, right? Like I was just like, yep, the car gets there by itself. I know exactly where we're going, you know? So as an 18-year-old, you're not paying attention to where, or, you know, between newborn to 18, you're not paying attention where the best grazing area, where the best food is located, because mom's taking you there, grandma's taking you there. But all of a sudden you get kicked out and you have to learn how to cook for yourself, do your laundry, clean, and no one has, if someone has been doing this for you all the time and you don't know how to do it, now you're the weakest one in the community, right? If you don't know where to find food, you have a higher chance of being predated upon. So modern elephants, to avoid being lion lunch, the young adult males will band together and create these loose gangs of associated teenage elephants, hence the Ninja Turtle. And for protection as well as for family support. So they come from this group of individuals, and now all of a sudden they're alone. So elephants are usually pretty gregarious and in a tight family circle. So they also need that companionship afterward until they get old enough and strong enough to be on their own. So the thought is that because these were the most vulnerable in the population, because they were fending for themselves for the first time, they had a higher chance of mortality. Great question.
Okay, so we are basically running out of time for the facility, so I'd like to, again,
Thank you.
Dr. Pirlo for a great talk tonight.
Thank you.
And the important thing for my students, the extra credit thing is up here in front. Thank you all. Have a good night. And please come again when we have our next talk on the 14th next month. Thank you.